Hi, this is Mr. Wartsky again, and today we're going to get into cells and prokaryotes, or as I commonly refer to them as Proks. Now, if at any time, please pause the screencast so that you can get the information down from the slides or draw pictures, but let's get going. So the first thing we're going to start off with is the cell theory. In 1805, Lorenz Oken took all the different parts of the cell theory and put them all together. So the first part is all living things are made of cells. That's one of the definitions of life. Two, cells are alike in structure and function. So a structure in one cell has the same function as the same structure in another cell. Cells need information in order to survive. That should be information, not into. That's my spelling mistake. Information in order to survive. That is in the form of DNA. And new cells come from old cells. That's the four parts of the cell theory. Now, but what this does not say is that cells need to be small. So why are cells so small? And we're going to do an AP lab on this. So cells are small because of two reasons. One, small cells permit specialization. So all our different cells have different roles, different functions in our bodies, make up different tissues. That's specialization. And it increases the surface area, that's what SA stands for, surface area to volume ratio. Now, the increased surface area to volume ratio allows cells to absorb more nutrients faster and get rid of wastes quicker. So our lab and activity will be based on this, of why are cells so small, specifically the increased surface area to volume ratio. So there are two types of cells. The first type of cell is prokaryote. Pro means before. Kary is nucleus, so it's before the nucleus. And eukaryotes, U means true, and karyote means nucleus, so it's a true nucleus. Now, for eukaryotes, there are two types of eukaryotes. They're the animal cells and the plant cells. Now, eukaryotes have structures called organelles, which means tiny organs, and they have specific functions. Examples of organelles I've listed there: the nucleus, the nucleolus, endoplasmic reticulum, mitochondria, and chloroplast. I'm hoping that you remember these from your freshman or sophomore year. We're going to talk about prokaryotes or Proks first, because they're evolutionarily the first cell that came along. They're bacterial cells. They're the simplest of all cells. You're going to have a picture of the next slide, and I want you to draw that picture. But we're going to write down the functions. So the structures that you're going to label are the nucleoid, and that is a naked loop of DNA. And if you take antibiotics, and anti means against, biotics means life, they're against life, they kill life. Antibiotics, chloramphenicol, nalidaxate, rifampin, all affect the nucleoid stability. If you have an unstable nucleoid, then that's bad for the cell. A plasmid is an extra small loop of DNA. Please don't confuse this with plastid, that's an organelle, but it is a plasmid. It's like a nucleoid, but smaller. It might contain genes, might not contain genes, not necessary for the life of the prokaryote where the nucleoid is. Ribosomes, you're going to see ribosomes in prokaryotes and eukaryotes. They have the same function. They produce proteins. They are part of protein production. And since the cell is the giant protein factory, ribosomes have an integral part in keeping the cell alive. Now, antibiotics, tetracycline and erythromycin, they prevent the ribosome from producing proteins, so that will prevent the prokaryote from dividing and allow our body defenses to kill that probe. Cell walls. All Proks have cell walls except there's one class that does not, and it provides shape and protects the prokaryote. There are two types of cell walls. There's gram-positive and gram-negative. And I'll go over the difference between gram-positive and gram-negative in class. Gram-positive has a thick peptidoglycan layer. Peptidoglycan is protein and carbohydrate. The gram-negative has a thin peptidoglycan layer, a periplasmic space which contains toxins, and a thin lipopolysaccharide layer. Lipopolysaccharide is lipid, which is fats, and polysaccharides, which are carbohydrates, surrounding the periplasmic space. And I will try to draw that and make a distinction in class. Now, antibiotics, penicillin and ampicillin, prevent the formation of the cell wall, specifically the gram-positive cell wall, which leaves the prokaryote exposed and allows the immune system to kill it. And here is a diagram of the prokaryote that I want you to draw. So put it on pause. Make sure you get all these different structures that are listed here, because I'm going to talk about some more structures on the next slide. The capsule. Not all prokaryotes have a capsule. This helps prevent desiccation, which means it helps prevent it from drying out. It helps the prokaryote stick to things, helps it move, protects the prokaryote. Prokaryotes with capsules are really tough to kill. There's some really nasty ones. The flagellum, which is singular for flagella, which is plural, but half of them have a flagellum. This helps it move by rotating. The flagellum in prokaryotes is a solid crystal, and it rotates like a propeller. Actually, this is one of the only examples of a wheel in nature. A pilus or pili, not all of these things or proks have a pilus, which is one, pili is many. These stick out, they're crystalline structures that stick out from the cell membrane, and they help the prokaryote stick to things as well as transfer DNA, and that's going to come in later. There are three main shapes of proks. We have a coccus, which is singular for cocci, and that is a sphere. A bacillus, which is singular for bacilli, which is a rod, and a helix, which is a spirilla or spirillum. Spirilla is plural, spirillum is singular, and it is a spiral. You'll see the main structures or shapes on this slide right here. They're underlined. We have a coccus, which is a sphere. There is a bacillus, which is a rod, and the spirillum, which looks like a helix, a corkscrew. So put on pause and draw those structures in the appropriate spot. So a cocci, why is it good to be a cocci? It becomes less distorted when dried out. And you're going to need to remember that when we talk about how things move in and out of the cell by osmosis. So remember cocci, specifically streptococcus, which causes strep throat. The bacillus increases the surface area to volume ratio, so allows it to absorb more nutrients faster. And the helix allows it to move better. If it has a flagellum, it can move in a corkscrew fashion through solutions, which allow it to move faster. So as we're talking about movement, we talk about chemotaxis. Chemotaxis is how prokaryotes move. If it's positive chemotaxis, it moves towards attractants like food. If it's a negative chemotaxis, it will move away from the substance. Those are repellents like poisons. So the description of the prokaryote movement can be described as twiddles or tumbles. I like twiddles. And runs. Runs are long swims, and if the prokaryote twiddles or tumbles, it basically stops and jiggles in a location. Why does it do that? Well, during the twiddles or tumbles, the prokaryotes are doing something known as temporal sensing, in which receptors bind to molecules. If the molecules are attractants, they will move to higher concentrations of the molecules. If the molecules are repellents, they'll move away from those molecules. So again, temporal sensing, receptors bind to molecules. If attractants, move closer to the higher concentration of attractants. If it's a repellent, we'll move to a lower concentration of repellents. Now here's a picture of runs and twiddles. You'll notice that we have a run here, which is a long continuous line, a tumble here where they stop and jiggle, then they move all over the place. And why do they move? Because they don't have eyes. What they're measuring is concentrations of the different molecules, which means or implies that somehow they can measure and compare one location to another location, which is pretty awesome if you think about it. So if you want to get that diagram, it might be a good thing to do. So How do some prokaryotes survive harsh environments? Some can produce something known as an endospore or a spore. These are basically bacterial seeds. So under harsh conditions, under high pressure, high temperature, low temperature, no oxygen, oxygen, a thick wall forms around the nucleoid and they will germinate under the right conditions. Bacillus anthracis does this. They form little spores. They go into the dirt that are ingested by other animals or breathed in by other animals. The Bacillus anthracis comes in nice, moist conditions. They will germinate and form the bacteria that causes anthrax. When they were having that problem with mail, people were mailing, or a person was mailing the spores from person to person. So we're going to talk about how prokaryotes reproduce. Now we have to define sexual reproduction, and asexual reproduction. So we need to define sex as a recombination of genetic material. Basically, when sex occurs, the DNA from two sources mix. That is sexual reproduction. Please don't confuse it with copulation, but sex is the recombination of genetic material. So when we have sex, the chromosomes in our gametes or sex cells cross over during meiosis. That means the DNA from two sources, the mom and the dad, mix. That is sex. We are mixing up the DNA from two organisms. Now, females or female individuals, before you're born, all 500,000 of your eggs will cross over prior to your birth, so you will not have sex anymore. Males, on the other hand, are constantly producing sperm, which are crossing over in meiosis, so males are constantly having sex. Prokaryotes, they do both asexual, which means without sex, without the recombination, without the mixing of DNA from two sources. Proks have asexual as well as sexual reproduction. In asexual, the DNA is copied, the cell splits, and a copy of the nucleoid goes to each cell. This is called binary fission. Now remember, fission rhymes with division, so that's what fission is division. Sexual, we are joining the DNA from two sources. That's how prokaryotes have sexual reproduction. And we'll talk about that in the next screencast. I know you can't wait.